Welcome to Chapter 18, which is the lecture all about infectious diseases affecting the cardiovascular system and to some extent the lymphatic system, which are two separate systems, but they kind of run and coincide with each other. So this is just a handy-dandy slide/image from your textbook, which kind of goes over all of the diseases covered in this chapter and even color codes them based on what type of microbe they are: virus, bacteria, protist, fungus, whatever. And so let's talk about some of these guys. So first, let's do a little bit of overview of the anatomy of the cardiovascular system and the lymphatic system. So the cardiovascular system is the heart and all the blood vessels, the veins and arteries and capillaries, and the purpose is to pump blood through the body, and the purpose of the blood is to really transport oxygen and nutrients to all the different tissues, and the heart is what keeps it flowing to all those different tissues. The lymphatic system is also a series of vessels that carries fluid, but the fluid is lymph fluid, and its purpose is to drain, is really drainage of liquid waste and an excess liquids from the tissue or water from the tissue. It also houses our immune cells, so a lot of white blood cells live there, and that is makes sense because as you drain fluids and stuff from the tissues, if there's any microbes there, then they go straight to these like lymph nodes, which are just packed full of white blood cells, which can mount an immune response. So it makes sense for the immune cells to really be housed there. The lymphatics do dump into the vascular system, so they are connected in that way. And you do have white blood cells that circulate in the blood, as you saw with your blood counts, the white blood cell count lab. So they are connected. Alrighty, so the cardiovascular system, a little bit more detail, the heart, the different parts of the heart. So if we look inside the heart is divided into 4 chambers. The atria are in the top and the ventricles are in the bottom. There's also valves that separate these chambers, and valves that separate the ventricle of the heart from the veins and arteries that leave the heart. So these valves, their purpose is really to control the flow of blood and keep it going in one direction so that you don't get blood sort of gurgling backwards. So it just keeps everything moving in a forward direction. But the valves, and you can see the valves are supported by these little tendons called the chordae tendineae, or sometimes just referred to as the heartstrings. And it's really the valves and these little tendons that tend to serve as surfaces that bacteria can stick to and grow on. So when we talk about endocarditis or bacteria growing inside the heart, usually the surface they're sticking to and growing on are the valves. The lining, the endothelial lining of the ventricles and the atria are really smooth and really difficult for bacteria to stick to. So the rougher spots are these, especially these tendons right here and the edges of the ventricles. So I just wanted to point that out. Right? When it comes to the blood vessels of the cardiovascular system, I'm going to cover up that word there, there's sort of 3 types. There's arteries, veins, and capillaries. So arteries always carry blood away from the heart. My mnemonic that I always remember is arteries away. They both start with A. And so usually they're carrying oxygenated blood that's been freshly oxygenated, and they're pumping it to the body. The arteries will branch into smaller arteries, which we call arterioles, which then will ultimately branch into capillaries. And capillaries are the smallest blood vessels, and they are where gas exchange occurs. So where oxygen leaves the blood to go into the tissue, and carbon dioxide leaves the tissue, goes into the blood to be taken to the lungs to be expelled. So any blood that's going back toward the heart is traveling through a vein. So therefore veins usually carry deoxygenated blood. The exceptions to that, whether they're carrying oxygenated or deoxygenated blood, is in the pulmonary circulation. So blood leaving the heart going to the lungs, that's an artery, it's carrying blood away, but in this case it's carrying deoxygenated blood to the lungs to get oxygenated. And then oxygenated blood from the lungs comes back to the heart through a vein, pulmonary vein. And so those are the ones that are sort of backwards in color coding in terms of their oxygenation status. So they also look slightly different in terms of their sort of physical structures. Arteries are very thick-walled, and they actually have a pretty thick muscular layer of smooth muscle. And so your arteries can actually constrict and dilate based on chemical messengers that cause constriction or dilation of that smooth muscle. And so that can, to some extent, control your blood pressure. Your blood pressure can be controlled by manipulating, your body manipulating the pressure on your arteries. Veins, though, have a very, very smooth or very, very, very thin muscular layer, and it doesn't. So blood flow through the veins is actually very dependent on the surrounding skeletal muscles. When they move, they actually sort of massage the blood through the veins. And also the veins have valves, which is something that arteries do not have, particularly veins in your legs. But it's just valves are present in any system really to help things keep flowing in one direction. That's really what a valve's purpose is. So these are your blood vessels. All of them have a very smooth inner lining, and so it is difficult for bacteria and organisms to stick to them. Remember, the first step of infection is adhesion. Once you get into the portal of entry, you have to be able to adhere and stick and grow, and that's difficult in healthy blood vessels, which have very smooth linings. But if you have a lot of inflammation or you have damage to a blood vessel, then the wall is kind of scratchy and it's rougher. And then things can start sticking to it, bacteria, but also blood clots and fatty plaque buildup. So keeping your arteries smooth and healthy is really the key to keeping a healthy cardiovascular system, preventing organic cardiovascular disease, but also infectious cardiovascular disease. The lymphatic system, made up of a series of vessels, we've talked about it before. You have the two organs of the lymphatic system are the thymus and the spleen, and then you have the vessels that carry the lymph, that the lymph fluid is drained, in one direction. So it doesn't circulate like the blood goes out to the body, back to the heart, out to the lung, back to the heart, out to the body. It's these two concentric circles. But the lymphatic system is all one-way flow. So it's all coming from the tissues to the lymphatics, then it dumps into the blood, and then any waste and stuff is sent from the blood to the kidneys where it can get peed out. And so the lymphatic system is always drawing from the tissues. It's not sending to the tissues. So there are some natural defenses of these systems. The cardiovascular system, we'll start there. First of all, it's fairly deeply located. So there's lots of layers of tissue protecting the heart. As you know, you got to go past the skin, and then you got to go past the bones and the muscles, and all the way to get into the chest. A couple of cavities, the thoracic cavity, and then another membrane layer called the mediastinum. So just by its location, its central location, that's keeping it protected. Also, the blood does have lots of white blood cells circulating, basically patrolling to keep things out. So, and you saw those all in your white blood cell labs. You saw all the monocytes and the lymphocytes and the neutrophils and the eosinophils and the basophils. And then there's also stuff you can't see, like complement proteins and antibodies that are circulating there. So lots of antimicrobial components in the circulating blood. If the cardiovascular system does get an infection, so if there is bacteria replicating in the blood, since the blood has access to the whole body, we call these infections systemic. A systemic infection is one that has spread to the blood and has therefore gotten around to the whole body. If the bacteria is actively replicating in the blood, that's called septicemia or sepsis, or sometimes it's called blood poisoning, but it's really not blood poisoning, it's an infection. Um, and and it's very dangerous because what happens is it triggers sort of massive inflammation systemically throughout the body, and that can cause damage to the blood vessels, to multiple organ systems, and that condition is known as septic shock. So it's characterized by fever, a massive drop in blood pressure, and organs start to fail. And the main thing is, one of the main things that they have to get under control in people with septic shock is that blood pressure, because you can't, if your blood pressure is too low, it means you're not getting blood and oxygen to all the tissues that need it. So that is the concern with bacteria in the cardiovascular system, in the vascular system, I should say, in the blood. So there are really no known normal flora of the cardiovascular system or the lymphatics. They're considered closed systems that are sterile. I think this is definitely true for larger organisms like bacteria and protists. I don't think there's any normal flora of the blood in that regard. There might be some viruses, though. There might be some viruses or phages that normally circulate in our blood, and we just haven't identified them because maybe they aren't harmful to us, but might be there. There's probably a lot of asymptomatic normal viruses. The virome is sort of next on the list. There are fewer people studying the human virome than there are studying the human microbiome. But I think that will be very interesting once we start learning that as well. We'll probably be surprised how many viruses we actually live with on a day-to-day basis. But for now, we consider the blood and the lymph to be sterile fluids, sterile compartments. Alrighty, so the first two diseases of the system that we're going to talk about are two of the world's biggest killers, the three global biggest killers of infectious disease are malaria, HIV, and tuberculosis. They kill more people in developing nations than they do in developed nations, as you can see from these charts. So this is a bit dated, but still, for the most part, holds true. This is the top 10 causes of death in low-income nations versus in high-income nations, according to the World Health Organization. So as we hear about in the U.S. as a high-income nation, we hear about cardiovascular disease being a major killer, and it is. Heart disease and stroke. Stroke is actually a form of cardiovascular disease. So those are the two biggest, that's the biggest killer, cardiovascular disease. Then cancers, respiratory cancer, colorectal cancer, breast cancer are all major killers, and respiratory problems, respiratory infections, COPD, chronic obstructive pulmonary disease, which is common in chronic smokers. Diabetes is a big problem, type 2 diabetes, type 1 and 2 diabetes mellitus. And Alzheimer and dementias, things that take the lives of elderly people. So we live to old age and then we get sick. That's a pretty norm in a high-income nation. Low-income nations, though, are much more susceptible to infectious diseases. So the only infectious diseases on the list in high-income countries are those respiratory infections like flu, pneumonia, bacterial or viral pneumonia. But here in lower income nations, respiratory infections are the #1 killer. And then diarrheal diseases, which are generally infectious. HIV and AIDS are infectious. Heart disease, malaria is infectious, not stroke. Tuberculosis, and then neonatal infections. So 6 of those top 10 things are infectious diseases, which is why they really are so important. So although we don't have malaria here, and we have controlled HIV with pretty good drugs, we still have really high numbers of HIV. We tend not to focus on them as much. We forget that they are such big global killers, but they are. So let's talk about malaria first. So malaria, it is, first of all, a protozoan. It's a protist parasite. It's not an amoeba. It's a plasmodium. So plasmodia move through, they have a movement called gliding motility. They kind of like move like they swim. And this is what they look like. These are red blood cells, and then these are some adult mature Plasmodium. So there's different species of Plasmodium that cause malaria, but the one I'm going to focus on, the main one that causes the most disease and the most morbidity and mortality, is Plasmodium falciparum. So that's the one we're going to focus on. It is the name malaria, interestingly enough, means bad air. So this is also a very old disease. The malaria, I think, is Latin for bad air. So this was like, very old language that was used to name this. So I suppose current conditions they still use Latin to name things, but still. It was originally thought that you got it from breathing bad air, like not fresh air, or from breathing air from sick people. They thought that it was an airborne transmission. Even before they knew about germs and germ theory, there was this belief that you get this disease from breathing bad air. But we know now that that's not. It's from an infectious pathogen, and you get that pathogen from mosquito bites. So malaria as a protist, remember, a lot of protists have multi-host life cycles. They need 2 hosts. And in the case of Plasmodium, they need 2 hosts as well. They go through one phase, the sexual phase of their life cycle in the mosquito, and they go through the asexual phase in the human. So it has to pass through both creatures in order to complete its life cycle. So in the mosquito, they reach sexual maturity, and they form zygotes, an egg, base first fertilized egg, which develops into several of these sporozoites, which burst out of this oocyst, and it's these sporozoites. So all of this happens in the gut of the mosquito, but then the sporozoites, they swim to the salivary glands of the mosquito, so that when the mosquito bites and injects some of its saliva in its human host, those sporozoites are injected into the human, and then they go into the first stage of the disease in humans is the sporozoites infect the liver, so that's called the liver stage. And then they go through sort of a molting process, and they burst out of the liver in a new form called merozoites. And the merozoites are the ones that then infect your red blood cells. And they infect a red blood cell, they mature in the red blood cell, and then they lyse the red blood cell and do it over and over again. So we're going to see that one of the symptoms of malaria, the key sort of feature of malaria, is these recurrent fevers. So like every 48 hours you have a fever. And those fevers coincide with the rupturing of the red blood cells. Some of these merozoites, after they go through this cycle, will mature into sort of sexually mature adult parasites. There's actually male and female protozoans, or plasmodia, and these are called gametocytes. And these gametocytes are what get picked up. Well, all of this stuff will get picked up by a mosquito that's biting, but it's these gametocytes that serve as the parent cells in the gut of the mosquito that make babies and then reinfect and continue this life cycle. So I have a couple of slides kind of showing this same process in different ways. So we've got the mosquito bites a person and gets these gametocytes. The gametocytes then undergo fertilization, sexual fertilization, and here's the zygote that forms, which is a fertilized egg, and it matures, and when it ruptures, it releases a whole bunch of these little sporozoites, which is what get injected into the human. And those sporozoites first infect hepatic cells, liver cells, where they transform. They go through like a molting, and they turn into this other form called merozoites. And the merozoites bust out of the liver, and then they cycle through erythrocytes, red blood cells. They infect a red blood cell, go through some stages, bust out of the red blood cell, infect more red blood cells, and just keep doing that. Some of those merozoites, some of the cells that rupture out, some of those red blood cells, instead of making more merozoites, some of them will make gametocytes. And those gametocytes are what are important for continuing that life cycle, and so on and so forth. But these merozoites can keep cycling through the blood like for a long time. So that life cycle that I just went through sort of twice on those 2 slides with pictures is listed out here for you. The mosquito, by the way, is a specific species of mosquito called, or genus of mosquitoes called Anopheles mosquitoes, and they prefer really warm tropical environments. So malaria is not endemic in North America because it's just not an environment where these Anopheles mosquitoes flourish. They can live in the southern states where it's warm, but we don't have any animals with malaria here, so the mosquitoes haven't picked it up. And luckily, mosquitoes haven't learned to board airplanes and ships and travel globally like we do. But there have been occasional cases of transmission of malaria in the US. Because people in the US, there are some people who have malaria that contracted it in an endemic country and come back here and potentially get bit by mosquitoes, but it's not enough, it's not a critical mass large enough to start like an epidemic in the US. But it is a huge problem in areas where it's endemic, where it's in the animals and the people, and there's tons of mosquitoes in a tropical setting. And it's very difficult to control because it's hard to prevent yourself from getting bit by mosquitoes. And there's lots of prevention methods we'll talk about to prevent getting bit by mosquitoes, but complete prevention is nearly impossible. So a little bit more about the clinical signs of malaria. So there is an incubation period. It's relatively long, a 10-day incubation period. So from the time you get bit by the mosquito, it might be 10 days before you're feeling any symptoms. And the first symptoms are like, prodromal, kind of like you're tired, you're achy, you might be nauseous and have a fever and chills. All right, but then it can progress and you start getting these fevers at these regular intervals, like every 48 or every 2 or 3 days. And those fevers are basically induced by the rupture of those red blood cells and the massive explosion of merocytes into the bloodstream before they reinfect new red blood cells. What's really dangerous, so this disease can cause anemia. It can also cause issues with blood sugar regulation because your red blood cells use a lot of glucose, and these parasites actually use a lot of glucose. So it can cause complications from blood sugar levels. But what's really dangerous and what kills most people, especially children, from malaria is cerebral malaria. And this is when the red blood cells, so one of the things that malaria does is it makes your red blood cells, the ones that are infected, start to look kind of spiky. And they become decorated with some of the proteins from malaria, and it causes those red blood cells to get kind of sticky. And they have a tendency to stick to blood vessel walls. And what's really problematic is if they start sticking in very small capillaries, particularly ones in the brain. And when they do that, then it blocks blood flow, basically causes like a clot in that spot, and then you can't get blood flow. So cerebral malaria is basically like a stroke that's caused by malaria blocking the blood flow to the brain, and it can cause coma and death. It's the major cause of death, particularly in children from malaria. So malaria as a pathogen has some unique features that allow it to be successful. One of the things it does is because it invades red blood cells. So like a virus, it goes into the cell. Not all bacteria do this. Some bacteria they infect the body, but then they sort of remain on the surface of cells. But malaria goes inside the red blood cells, and so they're kind of hiding there. And red blood cells are pretty defenseless as cells because if you remember, they're the unique cell in the body that are non-nucleated. They don't have a nucleus. So they basically don't have a brain to tell them what to do if they get infected. There's no, if a skin cell gets infected, it can make interferon. It can make things it has a nucleus, and it has genes that it can turn on in order to defend itself, but not red blood cells. They're pretty dumb packets of membrane and hemoglobin. And so the parasite gets in there and it's got sort of a free hiding place to grow and replicate. One of the things that lysing red blood cells do release, though, a cytokine they do release, are pyrogens, and that is what leads to the fever, these recurring fevers. Another thing that's really clever about Plasmodium falciparum is that it can do antigenic variation. So it has proteins on its surface, antigens on its surface that the immune system recognizes and makes antibodies to and makes killer T cells. But then while it's hiding out in the red blood cell, it can switch. It basically can change its disguise. It's like, a criminal who escapes, dyeing their hair, changing their hair, changing their eye color so that authorities can't recognize them anymore. And that's what this Plasmodium can do. It changes its surface antigens to different shape so that when it comes out of the red blood cells, all those antibodies that you've made against it are now just bouncing off. They don't recognize it anymore. So you have to mount an entirely new immune response to them. So this antigenic variation is what really allows it to escape the body's immune response toward it and not be able to beat it. So the transmission of disease, like I said, is due to the presence of this vector, which is the Anopheles mosquito. As you can see from this map, the distribution is really in these yellow places. So a little bit in Florida and Texas might be warm enough. You can find some of these Anopheles mosquitoes, but really mostly South America, Africa, Southeast Asia, Southern Asia, and the Middle East are where we're going to find these mosquitoes, and therefore where we find Plasmodium, malaria. So if you travel to any of these regions, you will probably be put on anti-malarial medications, which I'll get to in the next slide. In these regions happen to also be in a lot of developing countries, countries with really high populations of people and high density populations, and lots of outdoors, like people's houses are made out of like mud. They don't have screens and windows and closed off to mosquitoes, so they're very vulnerable to getting bit by mosquitoes. So there's approximately 300 million new cases every year of malaria and about 1 million people, and it's mostly children, that die from it. So that's a lot. And people can get reinfected. There's no lasting immunity to malaria. There are plenty of people trying to make vaccines for it, but because of that antigenic shifting, it's really tough. It's a sneaky parasite. So there are cases in the US, but most of them occur in people who they acquire it during travel in other countries, and then they come to the US, and then that's where they get diagnosed. So, alrighty, so how do you diagnose malaria? Usually through a blood smear, looking at the blood cells. So these are all red blood cells, and normally you don't see anything in red blood cells, right? They're just these empty packets that are kind of like reddish, pinkish things in the background. But when there's malaria there and you stain the DNA, you can see these little parasites inside the red blood cells. So there's different stages of growth. This is called the ring stage, the ring trophozoite stage. So it looks like a ring, like a engagement ring. This is a more mature stage, the schizont. That's right before the cell ruptures. And these are just uninfected red blood cells. So you can see very clearly on a stained blood smear, you can actually see the Plasmodium parasites, and they really jump out at you because normally there's nothing showing up inside red blood cells, and these little ring shapes are very unique. You don't really see that in any other kind of cell. Another way to test for it is doing serology, so looking for antibodies that the patient is making towards malaria. All right, so how do we prevent malaria? Well, there is no vaccine for it. There are drugs that you can take, and a lot of travelers will take them as prophylaxis. So you take like anti-malarial medications when you travel to malaria endemic regions so that you don't get infected. Those same drugs can be used as treatment for malaria as well, but better to use them as prophylaxis first. You can also, of course, do your best to prevent getting mosquito staying wearing bug repellent, using bug nets, staying inside, wearing, full coverage clothing, whatever. Those are ways that you can prevent, that are also prophylactic prevention techniques. Another really popular method is to just try to control mosquito populations. So actually a huge, we almost, I don't know if I should say we almost eradicated malaria, but DDT was a very powerful insecticide that was used in like the 1950s, 60s, 70s. And then Rachel Carson wrote this book that was all about how DDT was killing like wild bird populations and really damaging the environment. So they stopped using it. But it was actually being used primarily to get rid of mosquitoes that carried malaria. And actually a couple of countries were able to eradicate malaria through the use of DDT. So there has been some talk of bringing it back in certain places because it's such an effective insecticide. And if we could kill off all the infected mosquitoes, then we could really hugely reduce the burden of malaria. A more modern day approach to mosquito control that's less toxic to the environment, and that some of you may recall having watched a TED Talk about. I think I removed it last minute, so some people watched it, some people didn't. Anyway, it was about using genetic modifications in mosquitoes to make the mosquitoes basically immune to malaria, and then releasing those genetically modified mosquitoes to start spreading. And so they could still, we'd still have mosquitoes in the ecosystem. They'd still bite you and be annoying, but they would no longer transmit malaria. So that has been promising. They've done this in some places with dengue virus, which is another tropical virus, and they found some success, some reduced transmission rates. So it does show a lot of promise. The drugs that we have to treat malaria are quinine-derived drugs, so the quinolones like chloroquine. These are the ones that have been in the news of late as potential treatment for COVID-19, though there have been some early trials that have not panned out. In fact, there are some that the trial has been canceled because the doses of chloroquine were actually killing patients. So chloroquine is a fairly toxic, like these quinine drugs and quinolone drugs are fairly toxic drugs. If you've ever traveled and had to take them as antimalarials, they often have unpleasant side effects. I had one friend who went to, when he was in law school, when he finished law school, a bunch of his friends, they went to Thailand as like a big trip to, like, celebratory trip, and they were all taking antimalarials, and he started suffering from weird like neurological symptoms. He ended up having to go back to the US early and go see a neurologist, and they were sort of long lasting because the drugs stay in your body for a while. So yeah, they're not so fun. Another one, a newer one, is artemisinin, which is actually a plant-derived compound, and a lot of times these drugs are given together. So one of the quinolones and an artemisinin together work best because there are some malaria parasites that are developing resistant to the quinine and the quinolone drugs. So that's problematic because we're going to be losing our ability the little ability that we have to treat malaria. Again, you can get recurring infections with malaria, so it's not really feasible or economical or advisable for like people who live in endemic region. If I travel to Thailand, I can take antimalarials for a couple of weeks to prevent infection during that travel period. But someone who lives in Thailand can't very well take antimalarials for extended periods of time because they're just kind of too toxic to take them as prophylaxis for a lifetime. So we don't have a great answer for prophylaxis, though a funny phenomenon of a natural sort of form or of genetic prophylaxis is the mutation for sickle cell anemia. Sickle cell anemia is a disease. It's a genetic mutation. We talked about it actually when we talked about genetics and mutations because it's a single point mutation that changes one nucleotide base and it changes one amino acid in the hemoglobin protein, but it causes the proteins, these hemoglobin proteins, to clump and form these chains, which then stretch out and elongate the red blood cell and gives them this sort of crescent moon shape, or what's called a sickle shape. And these sickled cells, and somebody who has two versions of the sickle cell mutation, they have two copies of it, then they have pretty severe anemia. A lot of their blood cells are sickled, and they have trouble transporting oxygen, and those sickled cells can get stuck in the capillaries and cause blockages, and that's often what leads to death, is like stroke or something from a blockage from those sickled cells. But if you have only one copy of the mutation, so you have half normal hemoglobin and half mutant hemoglobin, you don't usually get much of the disease. So you don't have a ton of sickled cells, and they mostly get filtered out by the spleen. But you do have resistance to malaria. And so sickle cell trait, the genetic trait, is more common in people who have ancestry in and malaria endemic regions, so South America, Africa, Southeast Asia. These are all populations of people that have a higher risk of sickle cell anemia, because it's actually adaptive for their ancestors to have that trait. Sickle cell disease is not adaptive, because they would probably have died before they reached breeding age, but having one copy of the sickle cell trait actually increased their survival because it made them more resistant to malaria. So sometimes diseases have benefits that we don't realize until we look at sort of the big picture. So I'm not sure that we should give everyone a sickle cell mutation to make them resistant to malaria, but these can be genetic clues that help us figure out unique treatments or unique prophylaxis for malaria in the future. All right, another major killer that we're switching gears from malaria now to HIV. HIV is human immunodeficiency virus, and that is the virus. The disease that it causes is AIDS, acquired immunodeficiency syndrome. So initially, you may be asymptomatic, or you may have pretty mild symptoms. A lot of common signs or early signs and symptoms of AIDS are fatigue, diarrhea, weight loss, but then you start to fall victim to opportunistic infections and opportunistic cancers. Remember, cancer control is also a feature of the immune system, and what HIV does is it actually infects your white blood cells, and that's how it makes you immunocompromised. So there are several diseases listed here that we call them AIDS-defining illnesses. These are illnesses that really only affect people if their immune system is so shot because they have AIDS. So one of them that we've talked about before is cryptococcus. Cryptococcus in the nervous system, so like meningitis from cryptococcus. We only see that in AIDS patients. So if somebody comes to the hospital, they may not have ever been HIV tested before, but if they have cryptococcal meningitis, very good chance they have AIDS, and HIV test would be one of the tests that would be run. Some other ones that are on here that are good. Burkitt's lymphoma and Kaposi sarcoma. Those are cancers that really only show up in people who are severely immunocompromised, aka have AIDS. So HIV infection is very interesting, and I think I drew this graph out a little bit when we were talking about ELISAs and doing testing for HIV. So on here we have, on the y-axis, the concentration of whatever so there's some of these are antigen concentration and some are antibody concentration. So the concentration is the y-axis. The x-axis is time after infection. So initially, when you're infected, we're going to follow this green line here. So the green line shows us the level of antigen. In other words, the level of the virus level in the blood. So over the first couple of weeks after infection, the virus, it starts infecting white blood cells. It really likes T cells the best. And it will replicate and just make lots and lots of copies of itself, and you'll get rising numbers of HIV in the blood. All right. But then it will start to taper off. The virus will no longer sort of burst out of cells and be circulating in the blood. It will mostly be hiding out in white blood cells at that point. So the viral load goes down significantly. So after about 2 months, it's really kind of difficult to detect viral antigen in someone. So if somebody was exposed, they thought that, they had a sexual encounter 6 months ago, that now they're worried may have given them HIV. If they get a test, an ELISA test, they're going to want to look for HIV antibodies. That's the orange line here. So in orange, we're seeing, of course, it takes about 2 weeks to start producing antibodies for any given infection. And so those go up, and they stay steadily up because you're constantly exposed to this virus, so the IgG levels are going to stay up, right? They're just not very effective. Those antibodies are there trying to get at the virus, but it's not effectively able to because the virus, A, hides inside cells, and B, it's constantly mutating and constantly doing that antigenic variation and changing its surface antigens. So the antibodies you made last week no longer work this week, and the ones you make this week don't work next week. And so this virus is constantly hiding from those antibodies, and it's hiding from them inside the T cells. And so if we look at this purple line here, what's happening to T cells is they're slowly dropping until at some point you sort of hit like a threshold and the numbers start declining much faster. That you have more and more virus infecting more and more of these T cells and starts killing them off at a rapid rate. AIDS, one of the things, the defining features of AIDS is having a T cell count of less than 200 cells per microliter. So when you hit that point, your immune system is so depleted that that's one of the defining characteristics of AIDS. And notice that when that T cell count gets below 200, the viral load starts to go back up again. In other words, the virus is like, it's a free-for-all because the T cells have been so decimated that the virus no longer needs to hide. And so the virus can come back out again. So it's a sort of a data heavy, there's a lot of information in this graph, but it's very informative. It's a very informative graph. All right, so what kind of a virus is HIV? It's unique in that it's what we call a retrovirus. Here we go. We're going to go down here. It's an RNA virus, but it makes copies of its DNA, or it makes copies of its genome by converting the RNA into DNA. And it does that using this unique enzyme called a reverse transcriptase. So all retroviruses, the thing they have in common is they all have this reverse transcriptase, which is a virus unique enzyme. It doesn't exist in prokaryotes or eukaryotes. All cellular organisms go from DNA to RNA to protein, strictly. But what these viruses can do is they can go from RNA to DNA, and then the cell can take that DNA, turn it to RNA and viral proteins. So another thing that the virus can do is it can make that DNA and then insert that DNA into the host cell genome and become a provirus, enter a dormant state where it's not actively replicating, but it is hiding in the cell and sort of biding its time. In doing so, when it inserts the DNA, it can cause a mutation that leads to cancer. It can cause genetic defects that cause the cell to start multiplying over and over again, which is what cancer is. And so that's why retroviruses also increase the risk for these different cancers. The HIV virus is enveloped. It has an envelope. It buds out of cells, so it gets this envelope. And it has different spike proteins. And these spike proteins mutate very rapidly. But they're also important to study because it's the spike proteins that actually recognize the white blood cells and bind to receptors on the white blood cells, and that's how they get in to the cells. So there are two receptors that this GP120 spike on HIV uses to recognize cells it wants to infect. For one, it recognizes the CD4 receptor. Remember, CD4 is found on T cells, T helper cells. So it's the T helper cells they really like to infect. And then the second receptor that it needs to bind to is something called the CCR5 receptor, which is a cytokine receptor that's found on a lot of white blood cells. And the interesting thing about this, kind of like malaria, is there's a bunch of people, just people, like 1% of the population or something, that have a mutation in the CCR5 receptor. They just naturally inherited a mutant CCR5 receptor or a deletion in that gene, and so they don't have CCR5 receptors. And it's fine, their immune system is fine without it. It's not like a totally critical gene. But what it does is it makes them naturally resistant to HIV. Now, HIV is a new virus. It didn't enter the human population until, I think they've traced it back to about 1910 is when they think the first case jumped in Africa from primates into humans. And then in the 1980s is when it hit the US. But so this was not selected for evolutionarily by HIV, but they find that people who have ancestry in Eastern Europe are the ones that have the highest frequency of this gene mutation, and they hypothesize that it might have played a role in the the Black Plague, the Black Death. Maybe it was bubonic plague. Yersinia pestis. Maybe it's all the same thing. Now I can't even remember, but I think they were all Yersinia pestis outbreaks. So Yersinia pestis is the plague, and it's a bacteria, and it still exists, but it definitely caused some really big pandemics and outbreaks in ancient history. And it also uses the CCR5 receptor. So people who survived those plagues were probably more likely to have the CCR5 mutation and then, passed it down to their offspring. And now their great-great-great-great-great-great-great-grandchildren are also more immune to HIV. So, kind of interesting how certain mutations can, like naturally that exist in the population or were selected long ago by something completely different, can be beneficial. And I'm just kind of digressing still here, but a few years ago there was a really controversial genetic engineering case with CRISPR. This Chinese scientist, I think this was in 2018 or 19, and this Chinese scientist he kind of went rogue. He didn't get permission to do this, but he genetically modified a couple of embryos to have this CCR5 mutation. So his idea was he was making them resistant to HIV. He was trying to breed humans that were resistant to HIV infection, which I suppose is noble, but he didn't test it at all in animals, and there was no approval to do genetic modification in humans. And he modified these embryos and implanted them into a female carrier, and she gave birth to twins who were potentially the first genetically modified humans. Which I have forgotten to follow up on, but I don't know if they've tested because they may or may not have been successfully genetically modified. They may have just been born without those genetic modifications. But that was his thinking. In his mind, the most important genetic modification to make to humans was to mutate their CCR5 genes so they would all be resistant to HIV, which is not the first genetic modification experiment that I think you should be doing in humans, but that's a story for another day. So this is just a little GIF, a little animation of the HIV virus infecting a cell. So there's the GP120 spike and the CD4 receptor, and then a CCR5 receptor. They both grab the spike protein, and now penetration occurs. So that sort of triggers the cell to internalize it, and the membranes fuse. So you get entry through membrane fusion. The capsid dissociates and releases or that's the envelope fusing. So that's the nucleocapsid right there, the capsid going in. And now you're going to have uncoating. The capsid opens up and releases the viral RNA, and the little pink dot is the reverse transcriptase enzyme that's going to turn that RNA into DNA. And now that DNA can be made into double-stranded DNA, and then it can enter the nucleus and get inserted into the genome of the cell. So that's the nucleus there. It's going to go in through a nuclear pore, and through another viral enzyme called integrase, it will insert itself there. And then it just hangs out quietly in the genome. We call that a provirus. So it's inside the cell, but it's not actively replicating. It's just sort of reserved a spot. And then later, that it can direct the cell to start actually generating virus particles from that new gene it has acquired of inside the cell. And so now it's actually generating virus particles. So HIV enters through the blood and through the mucous membranes. So it can enter the skin via a cut, like an opening in the skin. And it can also enter through mucous membranes, the mouth, the oral membranes, vaginal and genital membranes. It will replicate in dendritic cells. So the dendritic cells are in the tissue, and they phagocytose things like HIV, but instead of digesting the HIV can actually replicate in the dendritic cells. And then dendritic cells go to the lymph node because they're antigen presenters, so they go there to present antigens to the T cells, but instead it's kind of like a Trojan horse. The dendritic cell is actually harboring all this virus and then delivers it to the lymph node where there's all the T cells that the virus can infect. And its favorite are the T helper cells, but it will even infect other types of white blood cells like monocytes and macrophages and B cells, but its favorite are those T helper, the CD4 positive cells, and that's the ones that really get decimated. So this viral production is still going on here. The viral assembly is taking place in this animation, and you see this budding, the budding of the mature virus particle. Ta-da! All right, so HIV transmission, like we said, can be transmitted through open cuts and through mucous membranes. Sexual transmission is very common. It can be transmitted through genital membranes. It can also be transmitted vertically from mother to fetus, and there have been a lot of improvements in birth protocols to reduce transmission. Another way that it's transmitted vertically is through breastfeeding. So HIV positive moms are usually recommended not to breastfeed. Unless they're taking HIV meds and their viral load is really, really low, then it might be safe, but especially if their viral load is high, it's not safe to breastfeed. But you can give birth to a baby. There are plenty of women who are HIV positive who have kept their viral loads low and were able to conceive and give birth to a baby that was HIV negative. But that's really mostly in situations where people have good access to good health care. Injections, so sharing needles through needle drug use is another really common form of HIV transmission. It used to be blood transfusions were a major cause before, like in the 80s and maybe even in the 90s, before HIV was really understood and its transmission was understood. It was a new outbreak. So in the 80s, what we are going through right now with coronavirus, like the world and the US was going through with HIV and AIDS, it wasn't understood. We didn't understand like the risk factors or how the disease progressed, and we've come a really long way. So now, of course, blood is always tested for HIV, even before you donate. So, so that's very rare now to get HIV from a blood transfusion. HIV doesn't live very long outside the body, so you can't really get it from contaminated surfaces or anything. You really have to have direct contact, direct transmission. So in that regard, it's not very contagious, and there's a lot of things you can do prophylactically to protect yourself. The more difficult is when you have two partners, one who's HIV positive and one who's not. So regular sexual contact, being able to protect oneself there, and then of course in vertical transmission, because these are people that are tied to one another. All right, so this is just another slide showing how HIV infects cells. It will infect the dendritic cell in the tissue when it gets exposed to the tissue, and then that dendritic cell will carry it in the blood, or the macrophages in the blood can get infected, and those are antigen presenting cells, so they travel to the lymph node to introduce antigen to T cells, but instead introduce the T cells to their killers, the HIV virus. So it's a hugely, it's been, I think it was probably the spread of HIV was due very in large part to mishandling of the infection when we first discovered it, because it was unfortunate enough to be a really big disease in a very taboo and discriminated population, which was gay men. It really, when it first was starting to show up in the American healthcare system in the 1980s, it wasn't called AIDS, it was called GRID. It was gay-related immunodeficiency is what it was called, because it mostly affected gay men. And that was a population of people that the government didn't particularly care about. And so they really didn't do much to prevent it, to research it, to figure out what was going on, until it really became an epidemic. And then more research started happening, more public education, but at that point it had already been spreading silently because it has a very long incubation time and a lot of people are asymptomatic. And so now the most common cause of transmission is actually heterosexual intercourse, not homosexual intercourse. So everyone and anyone can get it now and does get it now, and most of the world is infected because everybody has sex. So the current estimates are something like 36 million people around the globe are currently infected. And but we have really good drugs now. So only about 1% of people who get infected, or sorry, only 1% of people, there are a lucky 1% of people who get infected but never actually progress to AIDS. And those, a lot of those are the CCR5 mutants, people who have that mutation, so the virus just doesn't infect them very well. They have some natural immunity, all right? But so having HIV doesn't necessarily imply that you will progress to AIDS. Most people will if they're not treated, but we actually have very good treatments now, drug cocktails that help keep the virus levels low and keep that progression to AIDS very slow or never. So again, when you're testing for HIV, a very common test are these immunological tests, an ELISA, or even one of these quick tests, those lateral flow tests that are like pregnancy tests where you just put a drop of the sample, it flows, and 2 lines is positive, 1 line is negative. And so you can get these very quick tests done. Usually they'll follow that up. If it's a positive, they'll follow it up with an additional test to make sure it's a true positive. But it's also important about the timing when you're infected, because these tests, they test for antibodies in your blood. So if you get tested too soon, like you just had sex with someone 2 weeks ago, and you think you might be HIV positive, this test might come out negative this week, but in 2 months come out positive when your antibody levels are higher. So got to think about timing. These are just some of the definitions of AIDS. So HIV positive doesn't mean you have AIDS. It means you're infected with HIV. AIDS is an acquired immunodeficiency syndrome. So you have to meet these criteria of immunodeficiency. So CD4 T cells below 200 cells per microliter, or being less than 14% of all your lymphocytes, or experiencing one or more of those AIDS-defining illnesses that was on the previous slide. So you can be HIV positive, you can meet criteria 1 here, but not meet criteria 2. So if you don't meet criteria 2, you don't have AIDS, you are just HIV positive. So we have definitely massively improved prevention and treatment of AIDS. There's no cure for it technically yet, but there have been some experimental therapies where they've done basically bone marrow transplants. They've killed all the white blood cells in somebody's body through radiation and then repopulated their white blood cells with ones that don't have any HIV. And it has worked in just a couple of individuals. So it's a very expensive, personalized procedure, so it's still in experimental testing, but is promising. We also have several drugs that are available and are often given together in cocktails. So a single drug, it's pretty easy for an organism to develop resistance to a single drug. But when you mix the drugs together and you inhibit lots of different parts of the virus or bacteria at once, then you can get a better response. So that's why we sometimes call these cocktails. A lot of these drugs are actually mixtures or combinations of 2 or 3 or 4 different drugs. And these drugs can keep people who are HIV positive free of AIDS for decades, for their entire life. But they do have to stay on those medications. It can also keep the viral loads so low that it really reduces their risk of transmission. And in addition to that, we also have a line of drugs for people to take prophylactically. So we call it pre-exposure prophylaxis. So if you have an HIV positive partner that you are in a serious committed relationship with and you don't want to be abstinent, you can use condoms, but you can also take prophylactic medication to prevent yourself from getting infected. I think it's called PrEP. So those are some of the advances we have. And the drugs just work by inhibiting different parts of the viral infection pathway. So there's a fusion inhibitor that prevents the virus from fusing and binding to cells. AZT, which was sort of the original drug in the 80s or in the 90s, I think, is a nucleotide mimic. So it basically inhibits reverse transcriptase. And then integrase is another viral specific protein. So there are some integrase inhibitors that prevent the virus from integrating the DNA into the DNA of the cell. So mixing these together really helps to prevent the virus from being successful, but not enough to cure someone of it, because there's still inevitably some virus that's dormant in some cell that you can't quite get rid of. Okay, so I spent a lot of time talking about malaria and HIV. They're important, all right? The rest of the diseases are also important, but I'll spend much less time on them. So endocarditis, that literally means inflammation or infection of the inner lining of the heart. And really what we're talking about is bacteria growing on the valves. And we sometimes call these vegetations clinically. They're called vegetations or growth on the valves. And they can cause physical obstruction of the valve, so the valve won't work as well because they're sticky and mucky and all covered in bacteria. But also little pieces of that bacteria can break off and then cause sepsis and or land in another tissue and cause a local infection there. So these vegetations are not good. So some of the signs of endocarditis, and endocarditis can be acute, where it's like all of a sudden you have really severe infection, or it can be subacute, where it's kind of long-term and low level. So the symptoms are not quite that severe. So they can be kind of mild, like fever, fatigue, joint pain, swelling, anemia, all things that could be due to other things. You wouldn't necessarily put it together as endocarditis. Another sign of endocarditis is something called clubbed fingers, where the ends of the fingers take on this sort of swollen shape, bulbous shape. And these lesions on the hands are also signs of endocarditis. I don't know why. I don't know why endocarditis leads to these lesions, but they can be a clinical sign of endocarditis. So the causative agents of acute endocarditis, so a really quick and sort of severe infection, the most common causative agent is Staphylococcus aureus. And you'll notice some of these other bacteria, because we've talked about them before. Strep pyogenes can cause impetigo, and we'll see it when we talk about the respiratory system. It causes strep throat, and strep pneumoniae causes pneumonia, but we also said that it can cause meningitis. So really, acute endocarditis is often like a sequelae of another infection. Though Staph aureus does have this ability to just kind of sneak like into body tissues that it shouldn't be in and cause infections. It's a sneaky little bugger. And so if it gets to the heart and takes root on the heart valve, it can cause acute infection in the heart. The most common portal of entry, though, for endocarditis is through the blood, so through injection, parenteral, direct injection into the blood. So through needles, like needle use, whether it's drug use or injected medicine, through heart valve surgery, so the heart is open during surgery, and staph from somebody's skin could end up contaminating the surgical wounds. So those are the ways that you can get staph and cause acute endocarditis. Subacute endocarditis, that is more chronic almost, that slowly damages the heart valves, is due to a totally different subset of bacteria. And these are bacteria that are usually found in the mouth. They're dental bacteria. And there have been a lot of studies that have shown there's a lot of correlation between dental health and cardiovascular disease. So people, I think something like, 98% of people with cardiovascular disease have also had a root canal or something like that. So they think the hypothesis is that bacteria from the mouth get into, like in a gum infection or in a cleaning where your gum starts to bleed, that some of those oral bacteria get into the blood through the gums, and then they can travel to the heart and cause inflammation, or just their presence in the blood can cause inflammation of the blood vessels that triggers cardiovascular disease. So to prevent this, it's really important to have good dental health. Make sure you keep your teeth clean, go for your regular dentist visits, your prophylaxis and cleaning, to make sure that you don't end up with heart disease. A really severe form of cardiovascular disease is when bacteria gets into the blood and you get septicemia. And so septicemia, so I've talked about bacteria kind of getting into the blood, and that doesn't always result in septicemia, because if the bacteria, just a little bit of it gets into the blood, but it can't replicate there, then your circulating white blood cells will take care of that little bit of bacteria. But if the bacteria gets in the blood and can replicate there, then your white blood cells go crazy in the blood, and they start releasing all this cytokines, which damage everything. And that is what sepsis is. It's a massive immune response to bacteria that have invaded the blood. And the signs and symptoms often come on very suddenly. They can start off as just fever, nausea, increased respiratory rate, and low blood pressure, which are also kind of associated with fever. But the really key thing with septicemia is a severe drop, like a massive drop in blood pressure, due to major vasodilation from all that cytokine release. And so the blood pressure just really drops, and that's often what is fatal. And controlling blood pressure is like a really important feature of treating septicemia. So some other things, so fever, extreme pain, or people will say they feel the worst they've ever felt, or they literally say, I feel like I might die. I feel like I'm dying. Like that's how bad they feel. They may even have like consciousness issues, sleepy, difficult to wake up, be very pale or gray, have shortness of breath. All right, and septicemia is an emergent condition, like they need care right away to stabilize them and bring them back to homeostasis and kill the bacteria with antibiotics. So it's really what is the main causative agent is bacteria. When you have a lot of bacteria in the blood, when they're gram-negative bacteria and they release endotoxin, it's the endotoxin that really triggers that cytokine response and leads to that shock, that septic shock. Gram-positive bacteria can do it too through different cell wall fragments, not endotoxin, but definitely endotoxin is problematic. The most common cause of septicemia is MRSA. But most cases of septicemia are bacterial, though 10% of sepsis cases are from fungal infections. And this is sort of something from this like National Sepsis Society, that when it comes to sepsis, healthcare workers are supposed to, it's all about time. Temperature, so fever, infection, signs of infection somewhere, like swollen lymph nodes. Did they have a cough before this? Like, usually infection precedes septicemia, some other infection. Several infections can precede septicemia. If somebody has MRSA, if they have pneumonia, if they had meningitis, if they have a urinary tract infection, all of these can progress to septicemia. Mental decline, so again, that like from basically cytokines disrupting the brain and consciousness, so they can be very like lethargic, sleepy, semi-conscious, and then they feel extremely ill. So I'll tell a story. This is getting long, but whatever. You can pause and finish it later. I had a very scary experience a couple of years ago. Not me personally, but one of my good friends, her 5-year-old he was fine. He's a rough and tumble kid. And he's playing around all day, and he started vomiting in the afternoon. She thought, great, he's got a stomach bug. A couple of hours later, but he was still like fine. But within a couple of hours, he had a high fever. He was like limp, like not moving. He was very not himself, and very ill. And in mother's instinct, just feeling like something was really wrong, she took him to the emergency room, and they took his blood pressure, and right away they were like, he's septic. We need to transfer him. They ended up airlifting him to Albany Medical Center, and he was in intensive care for several days. It was really very scary. His blood pressure was so low, he had to be intubated. He had like multiple IVs. He was getting antibiotics, and he was getting like 4 different blood pressure medications, and his kidney and liver enzymes were high, and they were failing. But every day they were able to like make little baby steps and improvements, and like one of the medications could be discontinued, and then another, and then another. And he did miraculously pull through. I mean, it was really miraculous, but it happened so quickly. And he didn't have any signs of being sick. Like when she took him into the ER, I guess they did an exam, and it looked like he maybe had an ear infection, but he wasn't acting like a kid he didn't express any pain in his ear. I mean, and I said he was 5, but I think he was 3, actually, at this time. So just a small kid. And it turned out it was staph. It was from staph, not MRSA, but just staph. And they have no idea how he got, like, there's these sometimes bacteria just sneak into the blood. And I've heard plenty of stories of people who had, like, they got a cut on like a rose bush, like a rose thorn, that then turned into septicemia. Like everyday cuts, and just little, or just some mild infection that you have can, if you're unlucky enough, develop into septicemia. It just takes a little breach in your tissues for that bacteria to sneak into the blood and then be able to multiply. So it's more common. I think the most number of sepsis cases occur nosocomially, in people who are already in the hospital for an infection or for a surgery, and then the bacteria get in because of that. So it's definitely something that people in like recovering in the hospital have to be monitored for. But it's the community acquired ones that are so scary, because they can come on so suddenly from such minor things. But it is treatable. The key is getting it treated very quickly, usually through IV antibiotics and through just controlling blood pressure and organ function and stuff until the body can kind of get a hold of itself and that shock kind of wears off. So a lot of times therapy has to be started before you can get a blood culture to confirm which bacteria or fungus is causing the disease. So usually the IV antibiotics that are given initially, the empiric therapy, is something broad spectrum. And then when they have culture results, they'll switch it to a more narrow spectrum antibiotic. But really, it's the body, it's that cytokine storm that's so deadly. So trying to manage all of the damage that the cytokines are doing is just as important, if not more important, than treating those bacteria. All right, another organism, the plague, is caused by a bacteria that we'll see called Yersinia pestis. But this is the bubonic plague, the map of the bubonic plague, which was in the there were several outbreaks throughout the 1300s. And you can see how it sort of spread throughout Europe. So this was 1347 down here, and then the mid 1340s or late 1340s, and then spread to, so from the dark orange to the lighter orange and yellow areas. So there were other historical outbreaks too. So this was the Black Death. The bubonic plague is the Black Death. And it killed a third of the world's population. Again, this was back in the day before people knew anything about germ theory. They had no idea that it was a germ that was transmitting disease. They thought it was like God punishing people. That's why it's called the plague. They thought it was like a plague from God, but it's not. It's a bacteria that causes an infection, and this bacteria is called Yersinia pestis, and this is a picture of it. It's not actually a gram stain. I think it's an acid-fast stain, and it's often described as looking like a safety pin. If you look at it when it's stained, they kind of do look like little safety pins because it's kind of stained on either end. It has this bipolar staining, and it's transmitted from, so a lot of rodents are carriers. They are reservoirs of this disease, and they can be transmitted to humans through animal feces or through biting insects. So it was really fleas that were transmitting this plague in the bubonic plague and black plague. So Yersinia pestis gets transmitted to a person through a bite, but it can also be transmitted person to person through the air, and also it can be transmitted through animal feces. You can breathe the animal feces, and that transmits the airborne transmission of it. So back in the 1300s, in these densely populated cities where there was like really poor hygiene and no like sewage system, and rats just ran around, and the rats had fleas and the people had fleas, all right, it spread like wildfire. So there are a couple of forms of the plague. One is the bubonic plague, which is when the bacteria infects the lymphatic system and causes lymph swelling. And the swollen lymph nodes form these like giant swollen lymph nodes, which are called buboes. So this is one showing a swollen lymph node in the groin. So that would be a bubo. So that's the bubonic plague. And it also causes swelling of the spleen. Airborne transmission is when the bacteria gets into the lungs and causes pneumonic plague. So it basically causes a form of pneumonia. So pneumonic plague is, we're going to talk about it more in chapter 19 because it's the respiratory form of the disease. The bubonic plague affects the lymphatic system, so that's why it's listed here. So these are pictures of some buboes. These like just swollen lymph nodes there. And it had a pretty high mortality rate. Still does, actually. Back in the day, it had a really high mortality rate when a third of the world died from it. Currently, the mortality rate is 15%. And yes, Yersinia pestis still exists. Not so much in developed countries, but in developing countries, it's still around. With any of these diseases that can go systemic, that is the real danger, is if the pathogen gets into the bloodstream. And so plague can also become septicemic. Both pneumonic and bubonic plague the Yersinia pestis bacteria can spread into the bloodstream and cause septicemic plague. This is incredibly fatal. So mortality rates are 30 to 50% even with treatment. So without treatment, you're going to die. But even with treatment, there's only a 30 to 50% survival rate. And a classic sign of it, and why it was called the Black Death, is because it caused necrosis and death of tissue, which caused a blackening, a visible blackening or darkening of the skin. So where does it exist now? There are some Yersinia pestis. There is some endemic Yersinia pestis in the US, but not up here in New York. So we're okay. And in terms of globally, we have very small numbers of cases of Yersinia in the US, but in parts of Africa, South America, and Asia, it can still be problematic. So the distribution is still out there, but the incidence of the disease is really low. There's not a lot of new cases in developed countries because we have good health care. But in developing countries, it's much more of a problem. So one of the things that is important about Yersinia pestis, the plague, is that it is a potential bioterrorism agent. We do not have a vaccine for it. It is treatable with antibiotics, but again, the earlier the better before it progresses and does too much damage. And it has a really small infectious dose. So it can be potentially very contagious, and it can be aerosolized, and which would just increase its transmission. So there is fear for a lot of these pathogens that they could be used in bioterrorism events, and plague is one of them. So even though there's not a lot of incidence of plague locally or globally, there's a fair amount of people who are doing research on it because there's funding for it because it's a potential bioterrorism agent. Tularemia is an interesting one. It's very uncommon, but it's sometimes called rabbit fever because rabbits are the main reservoir for this bacteria called Francisella tularensis. And it has a very low infection dose. So just a couple of cells can make you sick. And so most people get it from rabbits, not so much pet rabbits, but wild rabbits, like if they hunt rabbits during the skinning process, or even veterinarians can get it. And it can cause like aches and chills and malaise, but it actually is pretty deadly. The death rate is as high as 30% because it can spread, infect the lymph, and infect the pulmonary system. So it can kind of spread more systemically, which is why it has such a high death rate. So it's really rare. It's only 100, 200 cases per year in the US. But because it's so rare, that also means that it can be mistaken for something else more common and mild, and then not diagnosed and not treated properly. And treatment is really important, like getting immediate treatment, because it is susceptible to antibiotics, but if you wait too long, then it will spread too much and become deadly. So there is a vaccine for it for people who are at risk, like veterinarians, basically. And also it was developed in part because, again, there's a fear of it being used potentially as a bioterrorism agent, because it is very easy to transmit. Another one that affects the lymphatic system is Lyme disease. So Lyme disease is usually not fatal, but it can cause a very long-lasting chronic syndrome that's similar to rheumatoid arthritis and causes joint pain. So in the beginning, so you get it from a tick bite. It's a bacteria, we'll see. And you get it from a tick bite, and the tick bite often, but not always, will cause this bullseye-shaped rash. And that is very indicative of Lyme disease, but it's not always present. Or sometimes it's in a part of the body that's covered in hair, and so you don't see it. And so missing this rash can often lead to progression of the disease without treating it. It can be treated very effectively in this early stage, but not everyone gets the rash, and everyone who gets the rash doesn't always notice it, depending on where it is. Other early symptoms are pretty mild: fever, headache, dizziness, and these could be from, oh, you skipped lunch. So if you don't get it treated, it can progress to second stage or third stage disease. So in the second stage of disease, you start getting cardiology, cardiovascular and neurological symptoms. You might develop facial palsy, where like half of your face goes numb and is paralyzed, and so you get this kind of funny looking countenance because half of your face is innervated and the other half is just droopy, like when you get a Novocaine shot for a cavity or something. And it's temporary, it goes away, but it's definitely can be a sign of Lyme disease. Once it progresses to that third stage, though, that's where it starts to really affect the joints. It causes really painful arthritis and also sometimes neurological issues that can be severely disabling. And these are chronic, and it's really difficult to treat Lyme once it reaches this third stage of disease. So the bacteria that causes it is called Borrelia burgdorferi, and it's a spirochete. We haven't seen any spirochetes yet. We've seen mostly cocci and bacilli, but this is a spirochete. It moves like a corkscrew, kind of. And it's transmitted by ticks, of a specific species called, or a specific genus called the Ixodes ticks. We commonly refer to them as deer ticks, and they're super, super small. So like this, for relative size, this is a dime. Dimes are really small coins. And like an adult deer tick is like barely bigger than the D on the dime. So still really small. The larvae are like, I don't even know if you can see them with the naked eye. They're so tiny. So I don't know how many of you guys have had ticks before, but like I grew up in the South, and the ticks there tend to be a bit larger, so like fairly sizable, easy to see. And my husband went out in the woods one time, and when he came back, he was like, "Can you check me for ticks?" And I was checking him, and there was like this tiny little speck in his armpit. And I was like, "Oh, I'm sure that's just a piece of dirt." But it was a tick, and I was surprised because I just didn't realize how small these little deer ticks are. So you definitely have to look carefully after you've been out in the outdoors in the Adirondacks, because Lyme disease is endemic here. All right, it's spreading in the Adirondacks, but definitely there's a high concentration of these deer and deer ticks in the Northeast, also in Michigan and Wisconsin area, and sporadically throughout the rest of the US, but really up here in the Northeast is where it's bad. So if you go out for a hike, which I encourage you to do, just make sure that you check yourself when you get back and check everyone in your household for ticks, because it takes a while for that tick to transmit the parasite. So the ticks like to feed on deer, and they actually get the bacteria from mice. So the larvae will feed on mice, and then they grow up and they live on deer, and then we tend to get it from, because of the availability of the deer in the region. So when we go out for a hike, we get a tick, we don't realize it, the tick bites us, and through that bite, it's releasing the Borrelia sporochetes, or spirochetes. Lyme disease, like malaria, can do that antigenic shifting. So that's how it evades the immune system and can cause these chronic infections. So it produces a lot of antibodies. You make a lot of antibodies, but the antibodies are constantly sort of being outgrown and being deemed ineffective because the spirochete keeps changing its surface antigens. So diagnosis can be difficult because of the range of symptoms is very broad. Different people will have totally different symptoms of disease. So it's been kind of hard to nail down the true symptoms of Lyme disease. Really classic signs are, of course, that ring-shaped rash, the erythema migrans is what it's called. If you can isolate spirochetes from the patient, if you can take blood and find spirochetes in the blood, great, there's a positive identification. You can look for Lyme antibodies, but I'm not sure all the tests for this are sensitive or effective. And you can also look for that spirochete DNA. So if you want to stay free of Lyme disease, either, stay away from ticks, but I don't necessarily recommend not going outside, but definitely try to protect, wear insect repellent, cover up, especially if it's not too hot out, to wear long pants and you can even like tie the pants at the bottom with like to keep anything from getting up through the sleeves of your pants. And then of course, when you get home, check yourself thoroughly for ticks, because if you remove them within the first, I think, 24 hours, then there's like no chance of them transmitting the spirochete to you. So it takes a while for that transmission to occur, for them to, it's like the last thing, I think, when they're done feeding, then they like squirt in some whatever juice, and that's when you get the spirochete. So if you can interrupt them while they're feeding, you don't have to worry about getting Lyme disease. And if you keep the tick, if you remove it and you keep it and you get it tested and it's Lyme positive, then you can take antibiotics to prevent yourself from developing the disease. So that's how you can prevent the disease. So this just is a picture of how to safely remove a tick. Another disease of the immune system is mononucleosis, or mono, what we call it for short, or sometimes you hear it called the kissing disease. And it's caused by a virus called Epstein-Barr virus, or EBV, which is a member of the herpes virus family. And remember, herpes viruses have that wonderful ability to go dormant and kind of stay with you forever and ever. I suppose the immune response to Epstein-Barr is good enough that you don't usually get this more than once, so it doesn't re-emerge. Once you have fought it off, it doesn't tend to cause disease again. The interesting thing about EBV is that something like 90% of people test positive for EBV antibodies, meaning they've been infected with Epstein-Barr virus. But a lot of them don't develop mononucleosis, this disease. A lot of people who get it, they just have like a cold. They have very mild symptoms, especially if they get it as children. It's just like a cold. But if you get it in your teenage years, for whatever reason, in the teenage to early adult years, there's a higher risk for developing mononucleosis, where you get really sore throat, swollen lymph nodes, swollen spleen. You may have hepatitis, inflammation of the liver, and you just get like very tired, a lot of fatigue, some atypical lymphocytes in the blood. So it can cause just really, and just kind of a little bit immune compromised because of all this inflammation in the lymph nodes and the spleen. So someone who has infectious mononucleosis, basically there's no treatment. There's no treatment for EBV. It's just treating those symptoms. So get plenty of rest. Avoid things that could damage your spleen. When your spleen is swollen like that, you definitely don't want to be taking any hits to the spleen. That's how, I thought that was how Houdini died, but actually I think his appendix ruptured. It's either his appendix or his spleen that ruptured when he got punched, and that's how he died. So anyway, if you have mono, he didn't have mono though, but if you have mono, you definitely want to, it actually can be kind of sore. You can have some soreness in the spleen. So definitely lots of rest. Take things to treat pain and fever until it gets better. It can be very debilitating when you have mononucleosis. And again, usually it's if you get it for the first time in your teenage years. So people who had it as a kid, and that's why I think it's called the kissing disease, because I guess it's around the age you start kissing people, and it can be spread through droplets, so you can spread it to people. But I've had students in the past who had full-on mononucleosis like when they were 9 years old. So you can have it. You can actually suffer from these symptoms outside your teenage and young adult years, but that's the age range where you're most at risk of suffering from the bad symptoms. A lot of college students get this. When I was in college, I had a lot of friends who got mono, and I've had students who had mono as well. And then lastly, if I haven't bored you enough yet, we'll talk about these hemorrhagic fever diseases. Hemorrhagic means bleeding, and fever means elevated temperature, and those are the two key characteristics of these hemorrhagic fevers. They're all listed here. That's something they all have in common. They all happen to be RNA viruses that are enveloped, and I don't know if that structure has something to do with the symptoms, but it's something they have in common. We'll talk about yellow fever, dengue, chikungunya, Ebola, and Lassa fever. The ones with an asterisk, yellow fever, there is a vaccine for that. It's only given if you go into regions that are where yellow fever is endemic. And I think we have Ebola vaccines in testing right now or in clinical trials. So we're very close to having an Ebola vaccine, largely due to the outbreak that happened a couple of years ago. I think it was 2016 or 17. There was a huge Ebola outbreak. Dengue, they're working on a vaccine, but it's proving very tricky. I'll tell you some interest. So, dengue fever, if you get infected once, that's bad. If you get infected twice, that's worse. The second time can kill you due to like exaggerated immune response that you have. That is actually not good. So trying to design a vaccine to it is proving troublesome because the second response upon challenge is actually what's so dangerous. Okay, so for none of these, I'll also say none of these have a treatment, an effective drug or therapy. So treatment is always just supportive care, trying to keep the patient alive until their immune system takes care of the virus. They all are hemorrhagic fevers. They all involve fever, and they all cause capillary fragility, which causes bleeding, internal bleeding, hemorrhaging. Okay, so the first one, most exciting one here, is Ebola and Marburg. Marburg is much less common, but they're very similar genetically related viruses. So Ebola virus is still endemic to Western Africa and Central Africa. It's thought to have come from bats, that it originally sort of evolved in bats but didn't make bats that sick. And then it was transmitted to other, primates and animals, and that it's transmitted to people because people in Africa, they're in much closer contact with wild animals, and they eat a lot of those wild animals for food, and so then they come into contact with them as a food source. And so I don't know when the first cases of Ebola arose. I can't remember if I ever looked that up, but I feel like it's relatively new, like in the 1900s it started. It's still considered an emerging infectious disease. This was the outbreak it was in 2014, or I guess that was longer ago than I thought. I thought it was 2016. But it was a huge outbreak. So all outbreaks of Ebola before that were maybe like, maybe 2,000 people got sick and maybe some, hundreds of people died. In this outbreak, 28,000 people got sick across 3 different countries in Africa, and 11,000 people died. So this is a pretty typical death rate, about 40 or 50% of case fatalities. And this was the trajectory in those 3 different countries, in Guinea, in Liberia, and Sierra Leone. So it was pretty huge, and there was some concern in the U.S. because we had a lot of CDC and World Health Organization epidemiologists traveling and doctors traveling to Ebola to help treat and control the spread of the disease. Control being the main thing, preventing the spread, just like we're doing social distancing here. They were sending epidemiologists to identify what was causing this rapid spread and how could they utilize protective equipment and train healthcare workers and train family members to properly handle infected people so that the disease would stop spreading, which it did relatively quickly because Ebola has a very small R0. It's not actually that contagious, not like airborne viruses are, because it's a bloodborne, mucus-borne virus. So you really have to like have direct contact with an infected person and contact their fluids and secretions. You can't get it from sharing air and breathing with someone. So some other hemorrhagic fevers that are in a lot of different tropical areas: chikungunya, yellow fever, and dengue fever. So if you travel to any of these regions, you will probably need to get a yellow fever vaccine. All of these, something they have in common is they're all transmitted by a mosquito called the Aedes mosquito, which is different Then the Anopheles mosquito, which transmits malaria. This mosquito also, by the way, fun fact for you, is the carrier of the Zika virus, which is not really covered so much in any of my lectures, but is teratogenic. It can cause birth defects. It's not a hemorrhagic fever, it just shares that same vector. So there are Aedes mosquitoes in the southeastern parts of the US, but we just don't have these diseases here anymore. They've been eradicated. Yellow fever was here in the late 1800s, I think. There was a big outbreak in Philadelphia, but it has been eradicated in the US. Still big in a lot of tropical regions. And these hemorrhagic fevers, again, they cause death basically through hemorrhage, through internal bleeding. They cause the capillaries to be just fragile, so you start bleeding. Another one, Lassa fever, is endemic to Africa as well, just like Ebola. And some of the cases are asymptomatic, but in some cases, about 20% of cases, people get really severe symptoms. And the reservoir are these rats that are often hunted for food. And you can get them from handling the rats. Or even if you step in the rat feces and you aerosolize it, and it causes a type of hemorrhagic fever. Non-hemorrhagic fevers, these ones are pretty uncommon, so I'm going to sort of rush through them. Brucella is actually more important of a pathogen for livestock. It causes these placental infections of livestock and causes death and miscarriage in a lot of livestock, and so it's really more threatening to the agricultural industry than it is to people. But people can get it as well, that if the bacteria enters through an opening in the skin or a mucous membrane, then it can cause disease, a disease that's called undulant fever. So it's a fever disease. Q fever is so rare. I'm just going to skip over it. Cat scratch disease you can get from cats that scratch you. Usually adult cats and not baby cats. And you can get it after, getting scratched or bit. And it can cause swelling of the lymph nodes and swelling at the site of the scratch. It's more prominent in kittens than adult cats. I had that backwards. So there's a lot of cases, most of them are in children, and it's totally treatable with antibiotics. But if you have kids and you have a kitten, it's something to look out for if they scratch your kid. Rickettsia is the organism. It's a bacteria that causes Rocky Mountain spotted fever. I still don't really understand why it's called Rocky Mountain spotted fever, because the Rocky Mountains are like over here and there's hardly any cases of it over there, but maybe the first case was identified in the Rocky Mountains. So it says it on the slide, Sarah. Read your own slide. It's named for the region where it was first detected, but it is not actually the region where it's most prominent. So it's a little bit misleading. But it's a tick-borne disease, so you get it from tick bites, and it does cause spotted rash, and it causes a fever, hence Rocky Mountain spotted fever. So we can add it to the list of tick-borne diseases, and it can be fatal. Actually, 20% of untreated cases are fatal. If you get treatment, antibiotics, because it's a bacteria, only 5% are fatal. So you get that distinctive spotted rash. So the name is helpful in telling you what to look for. But it can also spread to other parts of the body. And it's featured here because it can cause cardiovascular disruption, blood clotting, low blood pressure, that's hypotension, and hemorrhage. So for a lot of these diseases, avoiding ticks is important. And then I think the last one here, and I'll finally shut up, is Chagas disease, which is caused by a protist called a trypanosome. So this is often called American trypanosomiasis. In Africa, there's something called African trypanosomiasis, or sleeping sickness, and that's a different trypanosome. It's a different species. So this species, Trypanosome krusei, and the America we're talking about is mostly South America. And And it's transmitted by this bug here called a rejuvid, or sometimes called a kissing bug. And it can also be transmitted through bloodborne, like blood transfusions, or through the placenta from mother to child. And there's actually tons of people who are infected with this because it causes sort of a subacute chronic disease, has a very long incubation time, and can be asymptomatic for a long time, and it's hard to treat. And so there's a total of 8 million people who are infected worldwide, and there's actually 300,000 people in the US. That's a lot of people in the US that have trypanosomes. So Chagas disease can cause, in the acute phase, in the early phase, you might have just, you're tired, you have a fever, you may have a lesion that forms at the site of the bug bite, but then the chronic phase of the disease, which can be asymptomatic for a lot of it, eventually you start finding these trypanosomes throughout the body. So this protist just kind of spreads very quietly and not causing any symptoms until it's damaged your heart and your brain and your intestines, and you start to accumulate a lot of damage in those organs. And there's no vaccine for it. There are antiparasitic drugs that you can take, but they're only successful really in that acute phase. And if they're taken in that chronic phase, they're less successful, and they can't reverse any of the damage that's been done. They can maybe only kill the trypanosomes at that point. But it's this sort of silent killer that you don't know you have until it's too late sometimes. And then, okay, I lied, anthrax is in here too. We've talked about anthrax already. It's a bacillus bacteria, a rod bacteria. It's found in the soil. You can get it in a wound, and then you get cutaneous anthrax. But it can also spread systemically, and it's systemic anthrax that's relevant to the cardiovascular system. So that is anthrax. We'll talk about pulmonary anthrax in a later chapter. So we talked about cutaneous, and so injected anthrax, which is pretty rare. But through any of these forms can ultimately result in septicemic anthrax. They can all spread. So anthrax, of course, is a concern as a bioterrorism agent. In fact, has been used as a bioterrorism agent. In 2001, there were attacks in Washington, D.C. Different government officials were sent mail containing anthrax spores, and 22 people actually got anthrax, respiratory anthrax, and 5 people died. So it is an important pathogen we have to worry about. There is a vaccine for it that's given to people who are at risk, but this has to be done pre-exposure. So those people who got those spores in the mail, it was too late to get the vaccine at that point. And there's 6 shots that have to be given. So it's definitely not given lightly to people who are not at risk. If somebody's been exposed, you can give them antibiotics to hopefully prevent the spread of the disease, and you can treat it with antibiotics, but still, it's still pretty aggressive disease, and you can still die from it. So now I will stop talking and have a nice day.